So, good afternoon, everyone. We're going to get started with workshop 2, and this is on equity and vaccine responses. Our first speaker is Fan Zhu, who's going to be talking to us about age-related differences in responses to SARS-CoV-2 and endemic coronaviruses. She's from the University of Bergen.
Good afternoon, everyone. From University of Bergen, Norway. And I'd like to thank the organizing committee for giving me this opportunity to present our work on the age-related differences of SARS-CoV-2 and endemic human coronavirus reactive antibody responses. And as we all know, there is a long list of respiratory tract viruses circulating in humans, and overall, people of all ages has relatively similar rate of exposure. However, the mortality rate is the highest in young children under the age of 5, as well as in the elderly above 80 years old. Different viruses do have different high-risk groups. For example, seasonal influenza virus have this typical U-shaped mortality rates. However, the H1N1 pandemic in 1918 has had an extra peak around 20 to 40 years old. On the other hand, the RSV mainly attacks young children under the age of 5. In the case of COVID-19, the elderly, they didn't have the highest rate of infection. However, they did experience some very disproportionately high rate of hospitalization and deaths. And then this trend held true through the multiple waves within the first year. Therefore, the elderly were very often prioritized in vaccination in multiple countries, although they were not often included in the initial clinical trial while the vaccines were in development. And to study in depth the age-related differences, we collected pre- and post-vaccination serum sample from 80 vaccinees who received mRNA vaccine, 3 doses. And the age span is from 27 to 98 years old. None of them had a positive infection before receiving the mRNA vaccine. In comparison, we also included 29 COVID-19 patients from whom we had the first time point bloods collected as early as 1 day post the positive RT-PCR diagnosis, so that we can, in a way, treat this first time point as a baseline-ish. It's not really a baseline, but that is the best we can do in our lab. And none of these 29 patients had SARS-CoV-2 vaccination within the first 12 months of their infection. And we measured the full-length spike-specific IgG, S2 domain-specific IgG, as well as neutralizing antibodies against the infectious ancestral strain. And on the x-axis here, we have the age of all the participants. And on the y-axis here, it's the geometric mean of the antibody titer of all the participants given at each given age. And the pale blue is the background, and then the medium ocean blue is the antibody titer again after the first dose, and then the dark blue is the antibody titer against after the second dose. As we can see that after the second dose, almost everyone has relatively good amount of full-length spike-specific IgG. However, we see a sharp decline in the S2 domain binding IgG as well as neutralizing antibody from the age of 80 up to 90 years old. There's a dip. And on the other hand, in COVID-19 patients, actually there's no age-related decline of antibody titers observed. In fact, older patients, they actually had almost always higher amounts of antibodies compared to the young patients, probably due to their prolonged symptoms or more severe disease. And after the boost dose, which is also the third dose, both the spike-specific and neutralizing antibodies were potently induced in all the ages. However, you can see there's still this dip of S2-specific antibody responses as well as neutralizing antibodies around the age of 90, which means that those individuals above the age of 90 were still left with suboptimal amount of antibodies even after the third dose. In the patients, however, the antibody waned a little bit because they didn't have any vaccination within these 12 months. But then the older patients still had similar amount of antibodies compared to the younger patients. And this impact of aging on the SARS-CoV-2 specific antibody responses can be more clearly visualized if we do some calculation on the proportion of individuals who reached certain antibody fold induction at each given age. The calculation is a bit kind of confusing. But then, for example, here, if the probability is around 0.5, which means within this age cohort, 50% of the individuals achieved the antibody fold induction, kind of the preset criteria is listed here. And first of all, we can see a clear trend that in vaccinees, this antibody fold induction probability decreased over the age. However, in the patients, this probability actually increases. And more importantly, compare between the antibody responses after the second dose versus after the third dose. As we can see here, there's a lift of this probability of antibody fold induction in the age group from 60 to 80 years or from 60 to 90 years old, like here. So after the second dose, they are often below 25%. And then after the third dose, they were lifted a bit and around 50-40%. So they were helped very much by the third dose. However, all the individuals above 90 years old, they were still left with suboptimal antibody responses. And next, we investigated on the cross-reactive antibodies against the endemic human coronaviruses. And as many speakers this morning have already mentioned that there are 4 strains of human coronaviruses, 2 from alpha coronavirus, 229E and NL63, and 2 from the beta coronaviruses, OC43 and Hong Kong U1. Of course, there were thousands, if not millions, of different strains, but these 4 are the model strains widely used in many labs, I guess. And given the similarity of the viral genome among SARS-CoV-2 and the human coronaviruses, it's not surprising to see that in COVID-19 patients, often we see increased amount of these human coronavirus reactive antibodies. However, there is still kind of ongoing debate whether these cross-reactive antibodies are associated with favorable outcome, for example, less severe disease, or actually, indeed, they were associated with unfavorable outcome. And the latest publication suggested there's a correlation between long COVID with increased amount of these endemic coronavirus-related antibodies. And in our study, we measured these cross-reactive antibodies using both ELISA and the virus neutralization assay. With ELISA, we measured the spike binding IgG against the spike protein from all 4 strains of human coronaviruses, 229E, NL63, Hong Kong U1, and OC43. In addition, we developed the virus neutralization assay using infectious human coronaviruses, NL63, as well as OC43. And then if we take a look at the what happened in the COVID-19 patients, indeed, we see significant increases of antibody responses against all 4 strains of human coronaviruses, which is in line with most other publications. And in the vaccinees younger than 80 years old, we didn't see any increases of antibody against alpha coronaviruses. In fact, there is sometimes a significant decline, potentially due to lack of exposure. However, against the 2 strain of beta coronaviruses, we did see significant increases in terms of the binding antibody as well as the neutralizing antibody titer. And the similar trends could be found in the vaccinees older than 80 years old as well, although the magnitude of antibody increase is slightly lower in this age group. And last but not least, we did some correlation analysis, and indeed in patients there was significant correlation between neutralizing antibody and the SARS-CoV-2 specific neutralizing antibody as with the human coronavirus binding antibody. And then these 2 figures showed here the significant correlation between the SARS-CoV-2 S2 domain binding IgG fold induction and the human coronavirus full-length spike specific antibody responses. In the vaccinees, however, there is only very loose association between the SARS-CoV-2 S2 domain IgG fold induction and the human coronavirus antibody increase. And then this loose association is slightly more significant in the vaccinees older than 80 years old and less so in those younger vaccinees. Okay. In conclusion, we found that the first 2 doses elicit potent spike-specific binding antibodies in individuals younger than 80 years old. And then the 3rd dose, which is also the boost dose in our case, potently induced previously low S2 domain binding and neutralizing antibodies in the elderly 80 to 90 years old, and less so in those above 90 years old, for which I guess more doses of vaccine are necessary. And then in terms of the cross-reactive antibody responses, we found out that the endemic beta coronavirus reactive antibodies were boosted in all the vaccinees, although it's a bit lower in the vaccinees older than 80 years old. And in the patients, not surprisingly, we see kind of potent elevation of both alpha and beta coronavirus reactive antibodies. And then lastly, we found that the S2 domain specific antibody increases correlated with the human coronavirus antibody responses in both the patient and the vaccinees of all age. Therefore, we propose that the S2 domain could be a candidate for future universal coronavirus vaccine candidates. And with that, I'd like to thank all the members of Bergen COVID-19 research group and also all the study participants, all the vaccinees and patients involved in this study, as well as our collaborators. And I'm happy to take any questions.
Thank you. Are there any questions for Fandu?
No.
I found it interesting that you showed that the antibody levels in the elderly were actually still pretty substantial after vaccination. But we've also heard through several talks here that how important the T cell response is, in controlling the severity of the disease. So, do you think that further vaccinations would really help in that older population, or a better T cell response, is kind of required? In other words, if we give them more vaccinations and we get more antibodies, is that really going to help since they already seem to have, substantial.
I guess for all the ages, both the antibody and T cells, they are definitely equally important. However, for the elderly, since they are very old and kind of fragile, so I guess kind of the less symptoms, the better. So kind of the T cell response would provide survival and also prevent hospitalization, but some elderly, they were so fragile, they wouldn't even survive hospitalization. In that case, I guess they just need more antibodies to kind of lower the symptoms as much as possible.
Gotcha.
Yeah.
Thank you.
Well, I'm going to just ask a question here. In the early days of mRNA vaccination, one of the signals that came from Norway was actually deaths in the elderly shortly after mRNA vaccination. And it was because they were very fragile. And the reactogenicity of the vaccine was resulting in more deaths than old age homes had previously. So I think to the question that you asked, monitoring safety is also important, and they dealt with it by making sure that there was greater care given to people when they were vaccinated if they were especially old or fragile, as you said. Last question.
Yeah. Hi, this is more a technical question. So I was interested in the reactive antibodies to the endemic coronaviruses. Like, did you test that in neutralizing assays? And if you did, was this a pseudovirus system or did you use live virus?
We did run the virus neutralization using infectious virus. Yeah, we established this protocol for NL63 and OC43 here. We also in our lab, we also have the protocol for 229E as well. And I do have a poster kind of numbered 2044. There's more information on how to do the neutralization assay if you are interested.
Awesome. I'll stop by. Thank you.
Yeah.
Thank you very much, Fanzu.
Thank
That
you
was a
very much.
really clear presentation. And our next speaker is Lobke Hansen from the University Medical Center in Utrecht. She's going to be talking to us about T-cell immune responses induced by vaccination in Down syndrome.
Yes, thank you for inviting me to present my work here. As said, I'm Lobke Hansen. I'm a PhD candidate at the University Medical Center Utrecht in the Netherlands. And I will tell you a little bit about the T-cell immune response in people with Down syndrome after SARS-CoV-2 vaccination. So Down syndrome is the most common chromosomal aberration worldwide, and it's caused by a triplication in chromosome 21. And this leads to multiple dysfunctions of the immune system, amongst which a dysfunctional thymus. And this dysfunctional thymus then has a decreased output of naive T cells, and subsequently this leads to an increased frequency of memory T cells in people with Down syndrome. And on chromosome 21, there's also multiple immune genes located, amongst which 4 of the 6 interferon receptor genes. And they are upregulated in people with Down syndrome. And this leads to an overall interferon-stimulated gene expression, which is upregulated, and thereby leading to a general interferonopathy. So they have chronic interferon signaling, and the people with the highest interferon signaling also have more autoimmunity. And these changes in the immune system, as well as other changes in their anatomy of, for example, their lungs and their airways, make that they are more susceptible to severe respiratory infections, such as SARS-CoV-2. So for SARS-CoV-2, we know that people with Down syndrome, if they are hospitalized, they have a longer stay in the hospital, so higher hospitalization rates and increased mortality. And that is why we set up the PRIDE study, because we also know from literature that people with Down syndrome sometimes respond less well to certain vaccines. We set up the PRIDE study to look at the SARS-CoV-2 vaccination in adults with Down syndrome, and we hypothesize that they respond less well. So in the study, we included both healthy controls and people with Down syndrome, and we included them at baseline, roughly a month after the first vaccination and after the second vaccination, blood was taken. And the people were vaccinated with routine care vaccination and received either mRNA, so Pfizer or Moderna, or the vector vaccination AstraZeneca. And at these different time points, we performed a subset of assays. So we measured antibody concentrations on all 3 time points. And we studied the cellular immune response only after 2 vaccinations, so at time point 3 with the AIM assay. And we also measured interferon gamma release with the interferon gamma release assay. So if you look at the humoral response, we've shown that on the left you see mRNA, on the right you see vector, that this is suboptimal in people with Down syndrome. So in green we have people with Down syndrome, in purple we have healthy controls, and you see that after each vaccination there's a nice increase in the antibody levels for vector vaccination on the right and mRNA on the left. This is both the case, but if you look after 2 vaccinations, so time point 3, this is decreased compared to the healthy controls. However, the clinical relevance of this difference between Down syndrome and healthy controls is not yet known, but we know for the mRNA vaccination that it is also correlated with age. So the older the people with Down syndrome are, the lower their antibody levels also are. So in the Netherlands, this already led to a prioritization of people with Down syndrome above the age of 40 for early booster vaccination. So to study the T-cell immune response, we used the activation-induced marker assay as it was described this morning. Also, lots, so I don't go into depth that much, but we isolated PBMCs and re-stimulated them with SARS-CoV-2 specific spike peptides. And we took along a background and positive control and used flow cytometry to determine the percentage of SARS-CoV-2 specific T cells. So these are the markers that we used. For CD4, it was CD137 and OX40, and for the CD8, it was CD137 and CD69. And for further analysis, we determined the fold change, so the percentage of AIM-positive cells after peptide stimulation divided by the AIM-positive cells in the background. And that is what is shown in the figures. So here you see the full change of the AIM-positive cells for CD4. And in green, again, we have Down syndrome, and in purple, healthy controls. And the lighter colors are the mRNA vaccinated individuals, and the darker colors are the vector vaccinated individuals. So if you take all vaccines together, there's no difference between Down syndrome and healthy controls for CD4, and we see similar results for the CD8 T cells. However, if we take the vaccine groups apart, we see something different. So here we first look into the healthy control. And if we then compare mRNA and vector vaccination, we see that vector vaccination is decreased, has a decreased T cell response compared to the mRNA vaccination. But interestingly, if you then look into Down syndrome in the bottom and we compare the 2 vaccination, we see that there's no significant difference between mRNA vaccination and vector vaccination. So if we then compare Down syndrome and healthy controls for the mRNA vaccination, this leads to a decreased T cell response in people with Down syndrome after mRNA vaccination, which is more pronounced in the CD4 compartment compared to CD8 compartment. However, if we then compare Down syndrome with healthy controls for the vector vaccination, it seems that Down syndrome responds better compared to the healthy controls, at least for the CD4 T cells. So for the CD8 T cells, we see no difference. So this could be an effect of sample size. For the vector vaccination, we have a lower sample size, but we are not completely sure of these, why we see this effect, and we're looking into this right now. So to further characterize the T cells, we also measured interferon gamma. And here we looked at interferon gamma positive T cells after this AIM assay. So here on the y-axis, you see the percentage of interferon gamma positive T cells. And if we then compare Down syndrome with healthy controls, we see that there's no difference for CD4 and CD8 T cells if we take both vaccines together. So here we only took samples if we had at least 100 AIM-positive T cells to further subgate into interferon gamma positive T cells. But if we look at only the mRNA vaccinated participants for the CD4 T cells, because there we had a big enough sample size, there might be a slight trend toward an increased interferon gamma production in Down syndrome compared to the healthy controls. And if we look at the interferon gamma production with a different assay, so this is the interferon gamma release assay, where we re-stimulated whole blood with SARS-CoV-2 specific antigens, we see a similar result. And here we have taken both vaccines together. You see that there might be a slight increased interferon gamma production in Down syndrome compared to healthy controls. And this is more pronounced in the mRNA vaccinated cohort compared to the vector vaccinated cohort. And this might be in line with the general interferonopathy we see in people with Down syndrome, that they have higher interferon signaling in general. So to conclude, if we look at mRNA vaccination, we see a nice antibody response in healthy controls, but this is decreased in Down syndrome. And in healthy controls, we see a nice increase of the AIM-positive CD4 T cells, which is again decreased in Down syndrome, which fits nicely with the antibody results. If we then look at the interferon gamma production, we see that this is nicely upregulated in healthy controls, but this is even higher in Down syndrome. And if we then compare this to vector vaccination, we see that vector vaccination induces lower antibody titers in healthy controls compared to mRNA, and this is even lower in Down syndrome after vector vaccination. We see a decreased T cell response in vector vaccinations if we compare this to the mRNA vaccination. And if we then look at Down syndrome, we see that this is increased compared to the healthy controls. And we see a nice interferon gamma production after vector vaccinations in healthy controls, similar to mRNA production. And this is similar after vector vaccination in Down syndrome. So we want to further characterize the vaccine-induced immune response in people with Down syndrome with neutralization assays. And we also want to look at TCR sequencing and want to study the effect of the booster vaccination and also the durability of the immune response in people with Down syndrome. And with this, we hope to gain more insight into whether specific vaccine types may obtain better immunity in people with Down syndrome to prevent severe disease. And with that, I would like to thank a lot of people, and I'm happy to take any questions.
Thank you very much, Lauke. Very clear presentation. Are there questions?
Hi, I have two questions. First of all, as you subdivide these populations further and start to see significance appear, do you maintain your statistical power, or you just sort of, I always worry when people break stuff down further and suddenly see significance that you're just going, you're underpowering your population. So have you checked for power?
No, so this is quite recent results, so I haven't checked it. So that's something we still need to look into.
And the second question, I just would hope you could educate me on how you consent these people with Down syndrome that, you know, I don't know if they're considered a protected population, but I don't know anything, so something you could just elucidate.
Yeah, no, that's a very good question. So almost all people with Down syndrome have a guardian, and they sign off on whether they want to participate in the study or not. And as with all clinical studies, they can drop out at any time point without any further notice. So yeah.
Thank you. Are there any studies that are looking at booster doses? And I know that, you know, vectored vaccines started to be used early, and then there was a switch to mRNA vaccines in many countries. So you might have people who got vectored vaccines first and then got an mRNA vaccine subsequently. So do you have samples or people with that kind of vaccination history to study?
Yeah, so we have quite a big group of people that got AstraZeneca for their first 2 vaccine doses, and then were boosted with either Pfizer or Moderna. So those we are planning on following up and compare to people that got the homogeneous mRNA booster.
Thank you. One more question, sorry.
I just want to know if there's any differences in innate immune responses in people with Down syndrome, and if you looked at that post-vaccination.
Yeah, so we haven't looked at it in regard with the vaccination. So, but we do know that there are also differences in their innate immune system. So I've looked a little bit at monocytes, and there we see that they seem to be more activated in general. But that's something we're still looking into, but not in regard with the vaccination. So now we're purely focusing on the adaptive immunity.
Right. Thank you very much. So our next speaker is Francesco Berlenda Scorza from GSK and GVGH. He's going to be talking to us about a strategic model and industry collaboration for vaccines against neglected diseases.
Because we're going to be talking about a bacterial vaccine. And I'll try to explain why I think it is connected to vaccinology during a pandemic. And the connection is antimicrobial resistance, as you'll see. So the work that I'm presenting today is carried out at the GSK Vaccine Institute for Global Health. This is a dedicated organization. It's about 100 people working entirely on a global health pipeline at GSK. It's located in Siena. And we have access to GSK technologies, but we can develop vaccines of global health interest. So it's quite a unique opportunity. It was aspirational in nature when it was founded in 2008. But now we have our first product under our belt, a pediatric typhoid conjugate vaccine that is now licensed to Biologically in India. And it is marketed as Typhibev and procured by Gavi, and now licensed in 12 countries and under licensure in 30 more. So we're using the same model to develop other vaccines. We're expanding the pipeline. We certainly have an interest in enteric vaccines. And so today I'm going to be talking about Shigella. And you'll see why in a moment. But before we do that, Shigella is, and the reason we're developing a vaccine is that this is a main cause of diarrhea in developing countries, along with Salmonella and rotavirus. And so that's why we're developing a vaccine for prevention of disease in children. But also it has increased the profile in terms of antimicrobial resistance over time. And this has been known for a while, but more recently, something has happened or is happening, we think. And at the end of last year and earlier this year, a number of reports have come out. And as you see, all spaced in the range of a few months. We had all these reports, mostly from the UK, state of Washington and France, and we suspect that this is tip of the iceberg. And reporting XDR Shigella, XDR meaning resistance to basically all antibiotics that could be used in the clinical setting to treat the infection. So this is becoming a serious problem, not just in developing countries. This is mostly happening with a strain called Shigella sonii, and other times with Shigella flex 2a, at least in Europe and the US, or at least for now. And so this was becoming interesting so much that in February, the CDC issued a health alert because they were recording these cases in the US. And as you can see from here, out of the total number of isolates from Shigella in the US, in 22, 5% were XDR. This is a significant increase from 0% in 2015 and a very steep upward curve. Which we don't know where it's going. We don't know the 23 data, but the trajectory doesn't seem great. And so this happened during the pandemic. Is it related or caused by the pandemic? That would probably be a stretch. Maybe it's not what I'm implying, but the next question would be, is the pandemic making this better or worse? And so is there a connection? And so again, there's a beautiful report from the CDC that looks exactly at COVID and impact on antimicrobial resistance. And it's very well developed. They look at all different pathogens, including Shigella, but many others. So this is more of a case study, really. And what is interesting is that, yes, antibiotic use has increased. 80% of subjects hospitalized with COVID receive antibiotics, sometimes for a good reason, suspected co-infection with pneumococcus or secondary infection due to prolonged medical device usage, etc. But this has caused a measurable increase in antimicrobial resistant infections. And also, more broadly, there's been a decline in the interest of AMR over mainly an interest on pandemic preparedness. And we don't have all the data. We don't even have 2023 data. what we know is that the global sales for antibiotics has gone up quite a bit, and we know this will be related, sooner or later we'll measure the consequence on antimicrobial resistance. So back to our story on Shigella. So what can we do about this? Well, we think one of the things we have to do is to develop vaccines for these ever more resistant pathogens. One problem is that people have tried before us to develop a vaccine for Shigella and they failed. So why should we succeed? And so we had to go all the way to develop a new platform technology to develop a vaccine for Shigella and other enteric disease and other gram-negative bacteria, really. And we call it GEMMA. You can also call it outer membrane vesicles if you like. But these are not the detergent-derived vesicles we use, for example, for meningococcus B. These are released naturally by bacteria, with the exception that we can manipulate the toll pulse system that typically stabilizes the membrane. We use a genetic mutation to increase the blebbing. And this is a significant increase. You have to imagine in our manufacturing strains, after fermentation, you would get a 1-to-1 proportion of biomass and OMVs. So it's a significant. These are particles that are really representative of the outer membrane. They look like VLP under a microscope, and we can do further genetics on them to display different antigens. And of course, for Shigella, the antigen we're interested in is the O antigen on the surface. These particles contain it. For example, that would be different in a detergent extracted. You would lose it completely. So here we have it on the surface. The next problem is that the O antigen is linked to the LPS, which is extremely reactogenic. So we had to solve that problem. So this is a story that, as you see, started over 15 years ago, and we're still working on it, and I think we're getting it right finally. So in terms of reducing the reactogenicity of the LPS, there are ways to do it, and you see a couple here. Basically, they all work in the same way, so long as you end up interfering with the synthetic pathway of the LPS, and you have a pentaacylated LPS rather than the full hexaacylated, you lose the binding to human toll-like receptors, and the molecule is not reactogenic anymore. What you see there is IL-6 secretion from PBMC isolates, and that's how we measure the decrease. But we've demonstrated this over and over in GLP tox studies and human clinical trials. So once we do that, the LPS is non-toxic, and so now we have a vaccine that has the antigen that is not toxic, and we can move forward. And so the next thing is that we got something profoundly wrong at some point. What we considered a first-generation vaccine for Shigella turned out to be not very immunogenic in human trials, and indeed not protective in a human challenge trial, not surprisingly. So we had to go all the way back and understand it. And the vaccine we were using had a specific mutation for the reduced reactogenicity, but that mutation was reducing the level of O antigen on the surface. And the measure of O antigen, we've measured in many ways, but the most simplistic way to look at it is this Western blot. So what you see in B was our first generation vaccine, not a lot of O antigen. And unfortunately, that cost us a number of years, but now we understand why, and we found a different way to reduce reactogenicity. And with this new way, what you see in C, this material has plenty of O antigen, in fact, as much as the wild type that you see in A. So we have now what we call a second generation vaccine with a similar technology that is, we believe, immunogenic. What you see on the right is a comparison on the first and second generation in red and blue, respectively, by ELISA and serum bactericidal antibodies. So this was mice. We thought we had it on the right track. So we went on, and what you see here is rabbits, but also importantly, we went from a monovalent Shigella sonnei vaccine to a 4-valent with components that you see there. It's sonnei, Flex 1B, 2A, and 3A. And these 4-valent, we also measured how it would respond individually and in a high valency combination. And we observed that even the high valency combination was responding with no interference, with very good titers by ELISA, but also by SPA to look at antibody functionality. We also looked at whether the response to the vaccine, so this is sera against the 4-valent vaccine, and we looked at whether it would induce SPA against the strains included in the vaccine. Those would be the first 4 from the left, but also in strains not included in the vaccine. And although not as potent, it was surprising to see that there's activity against strains that are not in the vaccine. The explanation would be that the antigen has some common components, and that's probably where we get broadly reactive antibodies. So all these data were published just earlier this month. These are all the preclinical data, and they allowed us to proceed to clinical studies again. And here is the last slide I have, and this is unpublished data on our first study with the second generation. What you see in orange is I'm presenting a snippet from the whole study. We're only looking at Shigella sonnei here, and this is reporting GMR, so increase of the response post and pre-vaccination. And at the top by ELISA, so antibody titers, and you see a 20-fold increase over pre-vaccination. And at the bottom by serum bactericidal antibodies, and it's a magnificent 42-fold increase over baseline. And in yellow, that was the first generation vaccine. That's the one that I mentioned wasn't as immunogenic, and we think we really solved that. So this study met all the primary endpoints and the secondary that we had on immunogenicity, and so we've now moved on to the next study. So we're now running a phase 2 study, two phase 2 studies in Kenya, including an age-descending study. We're now in children 9 months of age as we speak, completed the first cohort there. We have 3 doses of the vaccine, and we've been able to up dose to the highest dose, and everything looks well so far. The study will read out in mid-24, and by that time we want to be ready to start phase 2B to measure actual field efficacy. So this is what I wanted to present as again, in context for what happens during COVID pandemic. There's also now an increased focus on antimicrobial resistance, and we're certainly Shigella is the most advanced, but we're applying this technology to other bacteria. The next one coming is an invasive non-typhoidal salmonella. We're using similar technology for it, and these proved their worth, and we'll expand it further. I just wanted to mention some of the people most involved in this work. At GVGH, so Francesca Micoli leads the program. Omar Rossi is the senior author on the preclinical study I showed you, and Usman Nakakana has designed the trials and is running them at the moment. So I'll pause there and happy to take questions.
Thank you for the presentation. Are there questions?
Hi, yeah, nice talk. I have a question. Well, this morning we heard that people tried vaccine candidates in the US, and then they went to developing countries and they saw differences in efficacy. I was wondering if you had any experience with this vaccine candidate.
Yes, so not yet, but we will see the results of the study in Kenya. What we know will be different is that adults in Europe are not exposed to this disease, whether in Kenya we know that there is pre-exposure, mostly in adults, and then when we go into children, we're not really sure. We have run an seroepidemiology study in children to see the baseline in the younger age groups. It seems that children are exposed to Shigella already by the first year of age, so that's why the vaccine needs to start very early. But yes, the behavior is very different, because if you're naive, you need really to induce a primary response, and otherwise what we would measure is a recall. So we'll have to see in children.
Thank you.
Thank you for the talk. I'm Matthew from Singapore. I'd like to ask, your deletions or I guess mutations in LPS, would they also be working for bacteriophages to detoxify LPS from them?
With bacteriophages?
Like with phage therapy.
Right. Yeah, that stretches my knowledge on bacteriophages. It's something that we can widely apply on LPS, and that binds TLR4 and needs to be exoacylated. It's pretty common in most gram-negatives or enterobacterial acid, but you can also find very different structures even in the gram-negative world. So it's not universally applicable, I would say. Yes.
Thank you.
One more question. What would be the use cases for a vaccine like this? You said this is for equity and essentially for looking at developing countries, but I could see from the descriptions of the epidemiology that you presented early on that this could be a dual-use product.
Yeah, so, I mean, over time, there's always been some interest for travelers and military, but with these outbreaks, we're trying to figure out whether there will be a wider use for this, considering these strains truly are untreatable, basically, XDR strains. And so I don't know that we have a solution. We'll stay focused on the pediatric indication now. We'd like to see efficacy there. One could branch out and do a human challenge, because that would be the right setting to see if we can see efficacy. I think we want to do that after we show efficacy in children. What we're also doing is a project called SHEMAB. We're looking at isolating monoclonal antibodies out of these studies. Not only vaccine studies, but human challenge studies and natural infections. And so that, I don't have time to present, but that's also a very neat piece of work where we have isolated monoclonals acting against on the O antigen most times. And we don't know yet whether it's applicable to the clinical setting, but at least we're developing the tools.
Okay, one final question for the young people in the audience. Why this model instead of the traditional academic model for discovery in universities and then translating to companies?
Yeah. There's probably 100 different models. We're not saying this is the only one, but this is one that we find has worked for us. And GSK, we have experience with different models. So GSK has developed Mosquirix, developing it all internally in the company all the way through licensure and then transferring it to a developing country manufacturer, to Bharat. That's one model. It wasn't very sustainable for the company to invest for the entire development. And it was done as part of the research of the company, so competing for resources. What we found by dedicating scientists to global health is that you don't have to compete for resources. And we think that the early developments where we can really build the product with the right technologies in, etc., that's where we really have a role. The late development, we think, should be done by the company that then has the product on the market. And so at the moment, this has been proven with the typhoid conjugate vaccine, although different products may need to adapt it, right, to the particular use.
Okay, thank you very much. So we are going to switch gears a little bit, and this is going to speak really to the topic for this workshop. Our next speaker is Jerome Dinger. He's a lecturer at the University of Buea in Cameroon, where he teaches molecular biology, biochemistry, and immunology, and his research is focused on malaria vaccine development. But today he's going to be talking to us about non-governmental, not-for-profit that he works with. He's going to present one of the projects of the foundation, the Michael Ganiham Geberad, I'm sorry about the pronunciation, foundation. Thank you.